Welcome back to the Deep Dive. Today, we're looking at something really cutting edge. It's right at the intersection of AI foundation models and, well, personalized cancer therapy. Our mission really is to unpack this fascinating collaboration between Google, DeepMind, and Yale University. Because they didn't just use a big AI model to crunch existing data. No, they used it to generate a completely new, testable scientific idea. And crucially, that idea was then proven right in the lab. It's a major lead for, potentially, new combination therapies.
Yeah, and this work tackles one of the really big hurdles in oncology right now, these so-called cold tumors. Basically, these are tumors the body's immune system just doesn't see. It makes a lot of immunotherapies kind of useless. If your T cells can't spot the enemy, right, they can't attack it. So the big goal here is finding a way to flip a switch, essentially.
Yep.
Make those cold tumors hot, visible.
Turning cold to hot. Sounds simple, but I imagine the biology is incredibly complex. So let's talk about the tool they used, because this wasn't just any AI, right? C2S Scale.
That's right, cell-to-sentence scale 27B. And it's not like an image model or something for protein folding. It's a new foundation model, 27 billion parameters, built specifically to understand the sort of language of cells. It uses Google's OpenGemma architecture. And, 27 billion parameters, that's seriously large scale for this kind of biological data.
We talk a lot about scaling laws on the show, how bigger AI models get better at writing or coding. But what does scaling mean here in biology for cancer?
Well, that was a core question for the researchers. Biological data is just exponentially complex, thousands of things interacting all the time. So sure, bigger models should get better. That's the scaling law. But they wanted to know, does a really big model just get incrementally better at old tasks, or does it gain entirely new skills, like the ability to generate genuinely new ideas?
So not just reading the book of life better, but actually understanding the grammar, the really complex interactions.
Precisely. And the specific bit of grammar they focused on was something called antigen presentation.
Okay, remind us why that's so critical for this whole cold to hot tumor thing.
Right. So antigen presentation is basically the process where a cell shows signals antigens on its surface. It's like waving a flag for the immune system. Specifically, you need these MHCI molecules on the surface displaying the tumor targets. If the tumor cell isn't doing that properly, the T cells just fly right by. The tumor stays hidden, cold.
Okay, I can see why that's a target. But finding a drug to force that sounds tricky. And this brings us to the next part, right? The conditional amplifier challenge. They weren't just looking for any drug.
Exactly. This is the really clever part. They gave C2S Scale a very specific, very conditional task. Find a drug that acts as a conditional amplifier, meaning it should boost that antigen presentation signal, but only if certain immune conditions are already present.
Oh, so it's not a magic bullet on its own. It needs a specific environment to work. Like it amplifies something that's already there, but maybe too weak.
That's the idea, precisely. The environment they specified had to have low levels of interferon. That's a key immune signaling protein. Enough to be present, but not enough on its own to really trigger strong antigen presentation. The drug needed to be like a catalyst, taking that weak signal and, cranking it up.
Okay, wow. That sounds incredibly difficult to screen for. A human researcher might just miss that, right? If a drug doesn't do much on its own in a standard lab test.
Well, yeah. Or even a smaller AI model. You need that sophisticated contextual reasoning. If low interferon is present, then drug X works. If no interferon, drug X does nothing or very little.
And that's where the scale comes in. That ability to handle complex if-then logic across messy biological data.
That seems to be the case. And to test this, the DeepMind and Yale teams came up with this method called the dual context virtual screen.
Dual context. Okay, break that down. How's that different from just, testing thousands of drugs in dishes?
Right. So traditional screening might test, say, 4,000 drugs against one type of cell in one condition, looking for one simple effect, like toxicity. It's kind of brute force. Here, they virtually screen over 4,000 drugs. But, and this is the key, they screen them against two totally different contexts at the same time.
Okay, what were the two contexts?
Context one was the positive one. This simulated real patient samples, so messy, complex environments with tumor cells, immune interactions, and crucially, those low levels of interferon humming along. Context two was the neutral control, just isolated cells in a dish, basically. No immune context, no interferon, completely decontextualized.
Got it. So the model had to find drugs that worked really well in the messy interferon-positive context one, but did basically nothing in the clean, isolated context two. That's how you find the true conditional amplifiers, not just general immune boosters.
Exactly. It's a tough conditional logic test. And the paper suggests this ability, this resolving of the context-dependent effect, really only emerged at the 27 billion parameter scale. Smaller versions of the model, they struggled. They could find drugs that boosted the signal generally, maybe, but they couldn't nail that conditional part. The only works if interferon is present bit.
That really suggests scale isn't just more power, it's unlocking a different kind of thinking in the AI, almost like creativity.
You could frame it that way. It's about synthesis, seeing patterns across vastly different conditions that might seem unrelated or too weak for a human or a smaller model to connect.
Okay, but I have to ask, they screened 4,000 compounds virtually? Isn't it possible a really meticulous human researcher doing enough experiments might have eventually found this synergy anyway?
It's a fair question. But think about human bias. We tend to follow known paths. If a drug targets protein X, we look for effects related to protein X. The AI, fed the language of all these cellular responses, isn't necessarily bound by those known pathways. It can spot correlations, however faint, across thousands of data points simultaneously. And the specific drug they found, well, let's get to it. Its effect in this context was novel enough that it really does suggest the AI synthesized something genuinely new.
Okay, let's hear it. After all that complex conditional screening, what did the model pinpoint?
It zeroed in on one specific compound, a kinase CK2 inhibitor called simotessartib, also known as CX4945. And the model was really quite insistent about it. It showed what the researchers called a striking context split. Big effect in the positive context, hardly anything in the neutral one.
Simotessartib. Okay, was this completely off the radar in cancer research?
Not entirely. The drug itself is known. It targets an enzyme, CK2, which is involved in things like cell survival, inflammation, general cellular stuff. But here's the novel part generated by the AI. It predicted a strong increase in antigen presentation, but only when simotessartib was used with that low-level interferon signal. People knew CK2 inhibition affected immunity somehow, sure, but no one had reported that this specific drug would boost MHCI expression or antigen presentation in this very specific conditional way.
Wow, so the AI didn't just pull a fact from a database. It connected dots that hadn't been connected before. It generated a new, testable hypothesis.
That's exactly it. Human intuition might have been focused on CK2's main jobs, maybe overlooking this more subtle synergistic effect that only appears under certain conditions. The model saw the pattern in the biological language.
Okay, prediction is one thing, proof is another. How did they actually test this in the lab? Did it work?
They did. The validation was really well designed. They used human neuroendocrine tumor cells. And here's a really important detail for, assessing how good the AI's prediction was. The C2S Scale model had never seen data from this specific cell type during its training. It was a true test of generalization.
Ah, so it couldn't have just memorized an answer related to that cell type.
Precisely. And the results. They completely backed up the AI's conditional prediction. Simotessartib alone, basically no effect on antigen presentation. Low-dose interferon alone, a little bit of an effect, modest. But combine them. A clear, significant synergistic boost in antigen presentation.
The conditional amplifier, it was real.
Yep, and they put numbers on it. The combination gave roughly a 50% increase in antigen presentation compared to the controls, which biologically means they achieved the goal, making that tumor cell significantly more visible to the immune system. The in silico prediction held up in vitro.
That is genuinely impressive. Okay, zoom out. Section 4. What are the bigger implications here, beyond this one drug combo?
Well, this is potentially huge. It's not just about finding simotessartib. This study provides a kind of blueprint, a new way to do biological discovery. C2S Scale found this novel, context-aware amplifier that gives a concrete, validated lead for new therapies against cold tumors. That's valuable in itself.
But it also changes the whole discovery process, potentially. Instead of scientists spending ages and fortunes on physical lab screens that might miss these subtle conditional effects.
They can use these large-scale AI models to run incredibly specific conditional virtual screens first. It shifts the paradigm.
From just high-throughput screening to maybe high-context screening, focusing the lab work where it's most likely to succeed.
Exactly. High context efficiency. It shows that training models on this biological language lets us uncover really complex context-dependent biology that was hard to see before. And the Yale teams are already running with this. They're digging into the mechanism, testing more AI predictions in different immune scenarios. It's just the start.
And critically, they've made the tools available, right? This isn't locked away.
Not at all. The model itself, C2S Scale 27B, the scientific paper preprint, the code, it's all out there on Bioraxov, Hugging Face, GitHub. It's basically an invitation to the whole research community. Here's a new tool, here's a new approach, let's build on this, let's speed things up.
It's an amazing story, from the problem of cold tumors through this massive AI to finding a specific drug synergy based on conditional logic.
Yeah, and if you think about the really big picture, the most exciting thing isn't just that AI can analyze data faster, it's this emergent ability we talked about, the ability to generate genuinely new, testable scientific hypotheses that maybe humans, with our biases and current methods, just weren't formulating.
So a final thought maybe for you, the listener. We often think science advances through human insight alone, but what if the next big leaps require this kind of AI synthesis? How many other hidden biological mechanisms, these conditional effects, are just sitting there, fragmented across published data, waiting for an AI large enough and smart enough to see the full picture?
Yeah, think about it. All those drugs that failed early trials because they didn't do much on their own. How many of them might actually be powerful amplifiers, just waiting for the right context, the right partner drug, the right subtle immune signal to unlock their potential?
It really makes you reconsider what discovery might look like in the age of AI and how these scaling laws apply not just to language, but maybe to the fundamental language of life itself.